Welcome to Lab 4, DNA Fingerprinting. In this lab, we're going to use suspect and crime scene DNA in order to solve a crime with gel electrophoresis. Before you even get into the classroom, your TA will provide you with microtubules full of suspect and crime scene DNA. Here you see 10 microliters of crime scene DNA being added to our first microtubule. Here you see us proceed to add 10 microliters of suspect 1, suspect 2, suspect 3, suspect 4, and suspect 5 DNA to each of the remaining tubes. If your lab has to add these DNA samples to your tubes yourself, make sure that you switch pipette tips between each one to avoid contamination. After 10 microliters of each DNA sample has been added, we're going to add this lyse solution. Make sure that the lyse solution is out of the ice for as short a time as possible, or else it will begin to denature. 10 microliters of our enzyme will be added to each vial. As you add your enzyme, make sure that you mix using the pipette as you were taught before. Once you're done pipetting your samples, you're going to take them over to the microcentrifuge. At the microcentrifuge, you should split your samples in two so that you can have a balanced microcentrifuge. You're going to close the top and pulse briefly in order to concentrate all of the liquid at the bottoms of the tubes. From here, your samples are going to be moved into an incubation fridge for the next 45 minutes at 37 degrees Celsius. Once your samples have finished incubating, you can begin to add them to your gel electrophoresis chamber. First, we will add our ladder solution containing 6 microliters of ladder and 5 microliters of UVU. While adding this, make sure that you keep your pipette stable and go deep enough so as to not release material into the buffer. In a repeat of our strategies from last time, we're going to add 5 microliters of UVU to each of our tubes. Once these samples have been mixed, they will be added to your gel electrophoresis chamber in the same way that was done last class. With each addition of UVU to a sample, you see UE mixing with the pipette 10 to 12 times. Once you begin adding your samples to your gel, make sure that you're pressing your tip deep enough, but not puncturing the gel. Also make sure that you're switching your tips between each time and pipetting deep enough so that you don't see streams of fluid coming out over the top of your agar gel.
And then it feels sad.
Yeah, it's just like, it's so sad. Aren't the players screaming at each other like, hey? Oh yeah, so there's a lot of... Does that bother you?
A little bit.
Samples should be in order, with your ladder on the far left, then your crime scene sample, then suspects 1, 2, 3, 4, and 5.
We pipette.
All the people that would sell food at the baseball games are now making money.
Yeah, but if a foul ball hits your cutout or one of like the pizza dude or whatever, they'll give you a foul ball. And they bring the announcers like pizza or hot dogs or whatever, you know? I know, I wish I was a baseball announcer instead.
Poor
I
you.
worked on.
Getting your PhD, not a baseball announcer.
Oh, I wish I was a...
Field later.
Oh my gosh, it's too late, Joe. I invested too much in this immortal jelly.
One of the world's leading poker players, she got her PhD.
Properly mixed samples will look like this.
She kind of...
We will again run these gels at 120 volts for 30 to 45 minutes. Once 20 minutes have elapsed, you can check the locations of your ladders to make sure that you're not going to overrun your gel if you go longer. If everything goes well, then the gel that you have should look something like this. Seems like we've found our culprit.